In this lecture, we have to talk about the inflammatory response, okay? And in the previous lecture, I told you that the inflammatory response begins right here in the tissues when macrophages, they would come in contact with some pathogen or bacteria, for instance. This red one is bacteria here. And then it is the job of an activated macrophage to inform all other cells of the immune system to come over to the site of infection and help with the job of the macrophage. The macrophage is bound to clear the pathogen, okay? But it cannot do this huge task alone. The macrophage needs the help of other cells. That's why the macrophage has to inform other cells. Now, what is the language of the macrophage? That the macrophage would speak to others and others would understand the message of the macrophage. When the macrophage has to deliver its message, the language of the macrophage is these different cytokines and chemokines that the macrophage actually is making and secreting these different cytokines and chemokines in order to tell other cells to come over to the site of infection. Okay? So these are actually, you can say, different words for communication of cells. The cells cannot speak to each other, okay, just like we do. So the cells communicate by using different cytokines and chemokines. Now when the macrophage has come in contact with this bacterium, it has sensed the presence of a bacterium. This presence is going to bring about lots of changes in the macrophage. The macrophage has sensed the danger and now it has started secreting and synthesizing different types of cytokines and chemokines, that there is a threat in the tissues and that you should come to the help of the macrophage. Okay? Different cytokines that we studied include different interleukins, like interleukin 1 family of cytokines, interleukin 1 and interleukin 1 alpha and beta. And I told you that interleukin 1 beta is the most important one because we call it the master regulator of inflammation. Okay? Interleukin 1 beta is preformed in the macrophage. The macrophage is filled with it already, you know? And there should be some way for this interleukin 1 beta to get out of the macrophage. Okay? So the macrophage can do it in two ways. One way is to secrete interleukin 1 beta, just like the macrophage would secrete all other cytokines, be it tumor necrosis factor, interleukin 6, or other chemokines, etc. And one way is to secrete these interleukin 1 beta cytokines, you know, in huge quantities. You know, we call it a burst of interleukin 1 beta, which is carried out by pyroptosis, the process in which, you know, some gateways or passages are made on the surface of the macrophage with the help of some proteins, and then interleukin 1 beta is just pushed out with the help of those passages into the vicinity of the macrophage. Okay? And in that process, the macrophage would also most probably die. It's not necessary that it would die, But in most of the cases, macrophage should die because the integrity of the membrane is compromised in that case. The entire membrane of the macrophage is just, filled with these channels or gateways that are made with different, with a protein. Now, interleukin 1 beta is needed for activation of the macrophage. It further activates the macrophage to be efficient enough to make and secrete pro-inflammatory cytokines, for instance, to make more of the pro-inflammatory cytokines and secrete them, because these are needed for different functions. And I told you that tumor necrosis factor alpha, for instance, is a pro-inflammatory cytokine. Because there is inflammation in the tissues, macrophage is activated, it secreted tumor necrosis factor alpha, and this is going to increase the permeability of the vessels. Interleukin 6 is going to induce fever response. Chemokine factor 8 is going to recruit neutrophils, and the chemokine factor 2 is going to recruit monocytes, to compensate for the loss of the macrophages at the site of infection. And interleukin 12 also is going to invite natural killer cells, which are the most important. You would just, you would go through the role of the natural killer cells as well. Okay? So all these things collectively are, all these cells are invited to the site of infection by the macrophage with the help of these cytokines in order to induce this state of inflammation. And inflammation is, the inflammatory response is needed to kill the microbe or to get rid of the microbe. Okay? So here you can see that what kind of chemokine is recruiting neutrophils. This is the chemokine factor 8 in the blue. Okay? This peptide or this chemical factor is synthesized here by the macrophage, and when it is secreted by the macrophage, this is a message for the neutrophils to join hands with the macrophage in the tissues. Okay? Now, how these neutrophils are recruited? What is the molecular process that is needed for the recruitment of the neutrophils? You know about the chemokine factors. There are 40 different chemokine factors made by different immune cells. And there are 22 different receptors for these chemokine factors on the surfaces of immune cells. If you look at the chemistry of these chemokine factors, they are either CC or CXC, CX3C or XC. There are 4 types of these chemokine factors. What is CC? CC is cysteine, cysteine. This is a motif found in certain peptides, cysteine residues. All this classification is based on cysteine residues. So CC would be cysteine, cysteine motif in certain peptides. CXC, this would be cysteine followed by X, which can be any amino acid, followed by C, which is cysteine. Okay? Cysteine, any amino acid, followed by another cysteine. This is another motif. Cysteine followed by any 3 other amino acids with a cysteine. This is another motif. Any other amino acid and cysteine, this is another motif. So these chemokine factors, when you sequence them and look at the amino acid sequences of these 40 chemokine factors, and you align them, or you blast them, you would find out that certain amino acids, they would have one motif, others would have another motif, but these cysteine motifs are there. So out of these cysteine motifs, this is CXCL8, okay? This is one of the second class you see here. This is a chemokine secreted by the macrophage, as you saw in the previous slide. And this CXCL8 recruits neutrophils from blood and guides them to the site of infection. This chemokine is needed for that function. This is a guide for who? For the neutrophils. It tells the neutrophils to come out of the blood vessels and go to the site of infection where the bacterium is. Okay? And for that process, the help of adhesion molecules are needed. Adhesion molecules would do adhesion. They are adhesive for the neutrophils. There are lots of neutrophils in the blood circulation. Neutrophils are normally in the blood. They are not in the tissues in normal situations. But when there is infection, the neutrophils are recruited from the tissues. They migrate from the tissues to the site of infection. So normally there are billions of neutrophils in the blood, and when there is some infection, the macrophages are activated there, and these macrophages secrete different cytokines like tumor necrosis factor alpha, interleukin 6. These are all pro-inflammatory cytokines. CXCL8, CCL2, interleukin 12. These are pro-inflammatory cytokines. So when cytokines are secreted, these cytokines are going to help in the neighborhood of the cells. They also have systemic effect. They are going to enter the blood circulations or act upon the endothelial cells as well. Okay? So when these cytokines would act upon the endothelial cells, if tumor necrosis factor alpha secreted by the macrophage would act on the endothelial cells, the blood vessels would dilate. Our permeability of the blood vessels would be increased. As you saw in the previous slide, that this is the job of the tumor necrosis factor alpha. When activated macrophages secrete that, it is going to dilate the vessels or increase the permeability of the vessels, which is needed in cases of inflammation, when there is inflammation. Okay? And the purpose is to increase the blood flow to the site of infection, so that more cells would get there to the site of infection to fight the pathogen or the microbe. Okay? So if one type of cytokine is just going to dilate the vessels, other cytokines are directly acting on the endothelial cells of the blood vessels, to express certain molecules on their surfaces, which are known as adhesion molecules. For instance, selectins, these are also adhesion molecules. Okay? This is endothelium, the blood vessels lining, internal lining. Okay? And this is the neutrophils within the blood vessel. So the cytokines secreted by the macrophage are going to act upon the endothelium, so that endothelium would express certain molecules that are not normally expressed by the endothelium in normal situations. Okay? These molecules are For instance, vascular adresin or CD34. These molecules are immunoglobulin-like molecules. We call them IKM1 or IKM2. Okay? These are selectins, we call them L-selectins. Okay? On the surface of neutrophils, if the cytokines act upon the surface of the neutrophil, it also is going to express integrin LFA1, for instance, on the surface of the neutrophils. Okay? So these are all adhesion molecules. These are ICAMs, immunoglobulin-like molecules, selectins, vascular adhesins, or integrins expressed on neutrophils, these expressed on endothelium. All these are going to entangle the neutrophil as the neutrophil passes through the blood vessel. Okay? The neutrophil this is a blood vessel, it's going through it. Okay? In normal situation when there are no hurdles, no adhesion molecules expressed by these cells, the neutrophil would freely float in the blood vessels because there are no hurdles. But once the macrophage has sent messages to endothelium and the neutrophils, now the endothelium has started making hurdles for the neutrophils because the neutrophil is needed outside the blood vessels, not inside the blood vessels. And for that purpose, it is needed that the neutrophil should be trapped here or lured here and taken out of the blood vessels. Okay? So there is expression of the adhesion molecules, and there are different types of glycoproteins or neutrophils that interact with adhesion molecules expressed on the endothelium. For instance, LFA1 binds with IKM1 expressed on the endothelial cell. Okay? Integrin LFA1 expressed on the neutrophil can bind with this adhesion molecule expressed on the endothelial cell. Okay? And the neutrophils, when these adhesion molecules are expressed, the neutrophil would start rolling along the endothelial wall. You know, the flow would not be free, it would be just rolling down. You know, these interactions are weaker when the neutrophils is binding with the endothelial cells using IKM1, for instance, or selectin ones. Then there is further bonding that you will see in the next step.
Binding for all the immunoglobulin, I can and selected that vascular adhesion important or just one kind defined.
Bonding of all?
Yes, binding of neutrophil to the endothelium. Does it require all the well-selected adhesions or I can or just one kind?
All of them are required for binding. In the next slide, I would show you that how the neutrophils strengthen this bond. Because this is not sufficient. If the neutrophil is still rolling down the surface of the blood vessels or endothelium, now it would need a bit of tight bonding so that the neutrophils can be fixed to one place where it can be just extravasated or taken out of the vessels. Okay? So there, the next step would be strengthening of the bonding between the neutrophils and the endothelium, which would ensure extravasation. The neutrophil would just go out of the vessels or be migrated out of the vessels. For migration, that step is needed. So this kind of bonding is just, creating small hurdles for the neutrophils, but when the neutrophils would bind with the endothelium using other receptors, then this binding would be tightened. Mhm.
There is often two types of cytokines, CC cytokine, cytosine X, any amino acid cytosine. इसमें जो हमारे पास X है, that could be any
amino
acid of
acids.
20 amino acids.
Yes.
So if it is adenine or guanine, अगर हमारे पास adenine होगा या फिर guanine, will they function differently या फिर अगर हमारे पास एक amino acid होगा कोई भी, तो वो एक चीज produce करेंगे.
Other amino acids don't matter here. The motif should be the same.
No
A
matter
chemokine would act, if it is CXC,
Yes, sir.
okay, all with this pattern, whether there is guanine or other amino acids, whatever is there. No, I'm talking about amino acids, not the other thing.
Yes, sir.
Okay, so if there is any one out of the 20 amino acids,
It's gonna be
then
the same thing.
it would act the same way. Yeah. Okay, this is now, you see the upper part of this slide is the blood vessels that you see in the red here. Okay. In the lower part, this one with these blue lines or patterns is the extracellular matrix. Okay. This is telling you how the neutrophil is coming out. So inside the blood vessels, this is a neutrophil. And you see the neutrophil has got different receptors. Like the CXCL8 receptor is already present there on the neutrophil. CXCL8 has receptors on different cells, including the neutrophil, including the endothelial cell, and including the extracellular matrix. Okay? It means the CXCL8 can bind to this chemokine factor, can bind to endothelial cells, neutrophils, and extracellular matrix. You would have heard about glycosaminoglycans. It would attach to glycosaminoglycans in the extracellular matrix. And similar glycans on the surface of endothelial cells right here, and it can bind to its own receptors on the neutrophils, which is the CXCL8 receptor. Okay? Now you see that these are adhesion molecules, ICAM1 for instance. There will be ICAM2, okay, which this is ICAM1, ICAM2, LFA1 in the case of neutrophils, okay. There is another receptor CD31 here that you see. On the surface of the neutrophil, here is a receptor CR3 as well. So you keep all these receptors in mind, okay? The CR3 receptors are normally present on the neutrophil. This is a complement receptor. And this complement receptor can also bind with adhesion molecules ICAM1. Okay? ICAM1 can bind to LFA1. Okay? The integrin LFA1 expressed on the surface of the neutrophil. And ICAM1 can also bind to the CR3 receptors shown here. ICAM2 also bind to this receptor. Now the CXCL8, this is the chemokine factor in green that you see here in different places. When the neutrophil is weakly bound with the blood vessels, when selectins or adhesion molecules are expressed on the surface of endothelial cells, then the neutrophil is now going to tighten its linkage with the endothelial cells right here. So chemokine factor is already there. It has been secreted by the macrophage and sequestered in the surrounding, where it has entered the blood vessel as well. Now the neutrophil is going to use its CXCL8 receptor to bind with the chemokine factor, which is already there on the surface of the blood vessel. Other adhesion molecules like ICAM and LFA1 are induced by the tumor necrosis factor alpha. I showed you that slide in which, the macrophage was secreting tumor necrosis factor alpha, and the tumor necrosis factor alpha was increasing the permeability of the blood vessels. Okay? The tumor necrosis factor alpha can directly act on the endothelial cells to express these adhesion molecules ICAM1 and ICAM2. It can also act on the neutrophils to express LFA1, which can bind with ICAM1. Okay? The tumor necrosis factor is coming from where? From the macrophage. It has entered the circulation. This is the systemic effect of the tumor necrosis factor now, that would act on the neutrophils and the endothelial cells to express these adhesion molecules. So the rolling neutrophil is now going to be engaged more tightly using the chemokine receptor here. The CR3 receptor also would bind to ICAM, and the ICAM1 receptor is going to tightly bind to the LFA1 expressed on the surface of the neutrophil. So this binding is now tightened enough for the neutrophil. So the neutrophil would be fixed to the site of CD31, which is another receptor. Okay? And now when it is bound there, this neutrophil is going to be dragged through the CD31 receptor. It would just give way to the neutrophils to enter from the blood vessel into the extracellular matrix. Okay? So now the neutrophil, with the help of these couple of factors, is going to leave the blood vessel and come through the CD31 receptors into the extracellular matrix. In the extracellular matrix, there are different proteins like collagens or laminins, and there are different proteoglycans like glycosaminoglycans. Okay? Now, the chemokine factor 8 is already there in the extracellular matrix. Because this chemokine factor had entered the blood vessels somewhere from the extracellular matrix, from the tissues. Okay? And I told you that chemokine factors have got receptors on the extracellular matrix as well. So they are bound there. There is, a gradient of these chemokine factors bound to the extracellular matrix. And if you look here, you can just see the gradients. There are less number of chemokine shown here, but as you go this way, the chemokines are increasing in number, the green spots that you see here. Okay? This is showing you a gradient. So the neutrophil is going to follow this gradient Where is this going? Where is the chemokine coming from? From the site of infection, where the macrophage is secreting these chemokines. So this is leading towards the site of infection, where the activated macrophage is secreting these chemokines. There will be more chemokines there in the surrounding of the macrophage. Okay? So this is going to follow this gradient and go there and join hands with the macrophage. Now, how is the neutrophil going to move in the extracellular matrix? Because there are different proteins, different fibrous proteins there in the extracellular matrix. The neutrophil is filled with different granules. The types of granules we already have studied when we were studying the neutrophils. And these granules have different proteolytic or hydrolytic enzymes. The neutrophil is going to secrete these granular components and digest the surrounding extracellular matrix. But would digest the surrounding, the neutrophil would be free to move in the extracellular matrix. Okay? It's not easy to move in this matrix. But if you digest these fibers that are crisscrossing each other, and the fibers are made of collagens and laminins mostly, then the neutrophils will be able to move along the gradient of the CXCL8. Okay? These are the granules in the neutrophils. So the neutrophil is going to secrete these granules. And these granules contain these hydrolytic enzymes or proteolytic enzymes that are going to digest these materials in the surrounding, and in this way the neutrophil would be free to move to the site of infection along this gradient. Now, how to make the neutrophils willing to migrate? Huh? You know, somebody should tell the neutrophils that you should, migrate from the blood to the tissues, These are the, you can say, from the outside there are some factors that are influencing, that are making, situation for the neutrophils to migrate from the blood vessels to the tissues, okay? The neutrophils also is undergoing certain changes from inside, Somebody should tell the neutrophils or convince the neutrophils to just, go out of it. It's better to go there and fight the battle because you are made for this purpose, So this is that kind of convincing. That is the story of convincing of the neutrophils, how the neutrophil is convinced. Against this factor is involved, the chemokine factor, CXCL8 is involved in this process, When CXCL8 binds with its receptors on the surface of the neutrophils, it has got a receptor that I showed you. On the surface of the neutrophils, there is a receptor for the chemokine receptor, chemokine CXCL8, and when it binds with its receptor, which can either be CXCR1 or CXCR2, okay? These are the receptors for these chemokine factors on neutrophils. Now, upon binding, there is some conformational change in the internal domain of this receptor, CXCR1 or CXCR2. And this is going to bind a protein which is known as a G protein. You know, this is a G protein. A G protein is a protein that is going to bind with a GTP, okay? Guanine triphosphate or guanine diphosphate. That's why we call them G proteins, okay? So once the CXCL8 binds with the receptor, conformational changes in the internal domains would take place in such a way that it would become perfect for binding with a G protein in the cytoplasm, okay, of the neutrophils. This is the story of the neutrophils that we are going. When it binds there, this G protein is going to bind with a GDP, guanine diphosphate with two phosphates here. Now if you see at the structure of the G protein, it has a big alpha part, and then there is a beta and gamma parts. There are three parts here of the G protein. And once this binding takes place, the GDP is phosphorylated, or it borrows another phosphate to become GTP, or guanine triphosphates in this pocket, you know. So this is a kind of activation for the G protein. The G protein would now dissociate from the receptor, and when it dissociates from the receptor, this also would split into two parts. The alpha part would become one, and the beta gamma parts would become another part. You know, it's split into two parts. And what are these factors going to do? These factors are going to convince the neutrophils to make more molecules of the kind, as you saw in the case of LFA1 induced by tumor necrosis factor, with the molecules that are needed for the extravasation of the neutrophils, the molecules needed for the neutrophils to come out of the blood vessels would be, you know, synthesized by the neutrophils once these messengers would act upon, you know, other proteins or activate other factors to synthesize the neutrophils. Okay? It is written here as well. You can also read this if you don't understand what I'm saying. That's why I've given this completely here. Okay? Each separate part of the G protein engages other proteins and delivers signals that collectively enable the neutrophils to leave the blood, enter the infected tissues, and hunt down the pathogens. Okay? What are the neutrophils going to do? Finally, they are going to help with the macrophages to hunt down the pathogen. The macrophage has started this entire situation. It has sent out messages to recruit different cells, and one of the cells were neutrophils, recruited with the help of chemokine factor 8, and now the neutrophil has undergone certain changes to be prepared for this situation to go out of the blood vessels and take on its job to help with the macrophages and kill the microbes, phagocytose the microbes, or secrete different kind of cytokines that would further enhance the inflammatory situation. Okay? So this is an addition to the neutrophils that we had studied last time.